about the kid endlessly. Sure, you're getting sick and tired of it. I know I am. Today, a couple of things to reinforce. We're going to get through bacteria and the bacterial cells, but we have to do, and the next step is we'll start to introduce and review aspects of you, because we have eukaryote to talk about in a little more, in a different way. We're not going to sit there and go, oh, chapter and verse of the mitochondria and the smooth and rough, because you know all that stuff. It's a mini review, but basically it's part of it. We have to understand that there's a number of eukaryotic, uh, microbiological organisms that have a great deal of relevance to us. I have an animation to show you, and today's topic du jour is really mostly about endospores. And there's an animation, and I just uploaded this to your and I'll do it obviously with the other group. It's a zip file. You probably can unload it. If not, you could certainly do it here and load a particular part of it onto your own if you did it on a computer here. So hopefully it'll be compatible. If not, I'll figure it out and load directly what I'm showing you, this animation. You can let me know if I need to do that. That's done. That's done. That's done. What am I doing? Okay, here we are. So we started, and let me get here. So, quick review. I'm doing this because I want to get in the same place with Trio in the back. You're not out of trouble yet either. Hagen is, but nobody else. So, it's okay. See, I'm starting to learn names. It only takes me a year to learn names. Some I can't. I already know. Yeah, right. Moving on. So, the whole idea of that, understand this idea of the DNA is very important because you have plasmids. Again, it's a circle of double-stranded DNA, a large one, enormous, or a comparatively small one. Okay. When we do genetic engineering, we add DNA we make in the lab, and we splice it onto a plasmid. We'll go into more of the details down when we do the genetics. And because these are non-essential pieces, they can come and go. They're useful for special traits that are useful to survive or to flourish in one way or the other. But understand that where we do genetic engineering, I'm emphasizing because I know it's on the test, and I send everybody the study guide. You probably saw that yesterday. Okay. E. coli and yeasts are used because yeasts are relatively simple when we're dealing with eukaryotic cells, and E. coli, which reproduce like crazy. And we have, and there's certain strains of both of these that are very receptive to donated DNA. And that's part of the process. And we'll look more of that into genetics. And I didn't get to that in the 11 o'clock group because I have another. It's hard. You're the first class here. They have a calculus class before, which sometimes runs a little over. So I tend to run out of time a little bit with that group that's there. So ribosomes we went over. Just remember that in the bacteria it's a 70S. Same with archaea. We're going to get to the distinction in the eukarya with regard to the ribosome. It's about density. Nothing to say here. Went over this. We use a lot of them again for identification, just to review. I went over all those taxes. That's important to remember. That's probably going to be on the exam. There's not much. I'm not going to ask about the cytoskeleton. There is one. We used to think there was not. And that brings us to endospores. They're so interesting, and they cause us problems because they're very hard to get rid of. We can get rid of the bacteria, but the endospores are constantly there. Okay. And again, for our purposes, they're almost always, we're going to be looking at two genera, two genuses, if you want to use the wrong plural, Bacillus and Clostridium, because I don't have a lot of information to share with you about Sporosarcina. Okay. And again, the big distinction, understand it always confuses it. That's why I'm reminding you again. The vegetative cell is a normal bacteria you're working with in the lab. You plated out bacteria. You saw how much they grew, right? So how those plates are filled with stuff. That's a vegetative cell. I'm growing. I'm happy. I'm vegetating. Okay, it confuses because in us, vegetative suggests that we're comatose and not going to survive. Whereas sporulation is brought on by adverse environmental conditions. Most of the time, that means not enough nutrients, not enough carbon or nitrogen containing materials that bacteria can ingest. Also, extremes in temperature and other lack of water, for instance, all of those may bring it on. But I think the major ones are carbon and nitrogen containing materials and inavailability of water. Living things, all living things, to our knowledge, require water to be able to survive. And they are just almost indestructible. And so I don't get a lot out of these, but I get a lot out of the animation. And it's one that I'll go over with you, and then I'll sort of narrate through it. And hopefully I'll try to adjust the sound to make your lives more amenable. And I did. Wait a minute, I got to check. I was free. I did start. So my wife tells me, and we'll look at that picture in a second. Here is this. So that is said, and so those are bacteria. So let's go from there.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Picolinic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cells, and the mature spore is released.
I'm going to go over it with you. Understand, when you do this in lab, you're going to see an array of endospores. Some will be within the cell, some will be already basically out of the cell. And when they say that they are nearly indestructible, it's absolutely true. They are extraordinarily long-lived, and the definition, and I may have probably told you before, and I'll tell you a bunch of times more, of sterilization is the destruction of all microbial life, including bacterial endospores. If you get that, everything else is gone that we know, that we can account. Prions are an oddball, we'll get there. And we're not doing that particular unit right now. So see what happens. These are gram-positive rods. You can see the peptidoglycan as part of the picture. Effectively, this is the big mistake that a lot of students don't see it. This is not a form of replication. They are not making more. And you'll see why in a second as I explain it to you back from the beginning. Oh. See, I could do that. And I'll bring it through this way. So when you take, you can do this without sound, if you don't mind. I got to play it.
Spore formation is initiated.
Just be quiet. Okay. So if you see this, you see a normal bacteria. They call that the mother cell. Notice it divides. Typical cells divide. This divides asymmetrically. When it divides asymmetrically like that, now you have the mother cell and what's called this forespore, F-O-R-E spore, which is basically the spore that's yet to develop. The mother cell, effectively, you notice the asymmetry is all about the vast majority. There's a little bit of cytoplasm here, enough to sustain this whenever the spore becomes active again. And so what we see happens here, and why it is not, here's the forespore, here's the mother cell. the mother cell's membrane, creating this sort of double membrane, envelops it. Why it is not a form of binary fission the way bacteria replicate is the other side, the other half of the DNA, the duplicated DNA degrades. So we're just changing from the vegetative form to the spore form. That's all it is, and it's all about the environment. Nothing else. And so what happens is we continue on with this picture. You have between those two membranes, we have the deposit of, as you can see by color, peptidoglycan. And now that combination of the membranes with the peptidoglycan embedded in it, sandwiched in it, is called the cortex. So already have a layer. Now inside, there's a couple of interesting chemicals. Think of it as dehydration. When you want to preserve something, okay, a lot of times, classically, you'll dehydrate it. We have one of those things at home, okay, a food dehydrator. If you want to preserve certain things, and you can buy them in the store. If you ever buy like packages of like mushrooms in the store that you find there, they're all dehydrated. You just put them in warm water, they rehydrate. We freeze dry things. And we'll go into different means of preservation as part of this course as well. Why? Because it involves food, and food is significant. One freaking smile would be nice. I know it's a Friday. And so what happens is dipicolinic acid attracts calcium, hardening the shell, and spits out water, dehydrating it. And then on top of that, we're not done yet. You get a protein coat that develops. And the protein coat develops, gives it strength, and sometimes there's an additional coat called the exosporium. And effectively, it's indestructible. Whatever they want to say, they've carbon dated these things ridiculously long. We're talking about, extra hundreds of thousands of years. And then when it's released, we have that. It's a very interesting phenomenon. So you can listen to this as many times as you would like. It's fine with me. And you can look through these. This doesn't help me as much as the other things do. But I thought I would share that with you so you can kind of understand the importance. And again, gram-positive, gram-negative, assembly of what peptidoglycan is made of, the nucleus stuff, the taxis stuff, endospores. That's the big part. If you looked at the study guide, that's the meaty part of this. Understanding what goes on in bacteria. It's mostly out of this chapter, these two bacterial PowerPoints that I'm using. The names we saw before, I don't have to go over. Okay. See, like for instance, why wouldn't it be the nucleolus? Because it's bacterial cell, it has to be a nucleoid. They don't have a nucleus, nucleolus. They don't have a nucleocapsid because it's a virus. Okay. They certainly don't have a nucleolus. Read the question. And look at all the answers. And we're not really doing the archaea. We know it's closely funny. They're more closely related to us. Archaea more related to us than bacteria because of certain RNA sequences and certain DNA sequences. Just take it. Okay. And again, the extremophiles, we've already touched on that, et cetera, and I'm not going into all that. This is lab. Okay. Basically, what you'll do in lab, like when we do in bacteriology lab in the hospital, you'll learn about something if you're going into a field where you're going to use what's called culture and sensitivity. You've got a wound. It's got yuck coming out of it. You get some yuck on a sterile Q-tip. And basically, they do what you did in the lab. They plate it out on different types of augers. And you do different tests to it. And basically, it's got a fancy, it's kind of a yes or no type of thing. Is it gram-positive or gram-negative? Does it have a capsule or not? Does it have endospores or not? Okay. They do this. And basically by this series of tests, is it ferment this sugar? Does it not ferment that sugar? Does it contain this enzyme? Does it contain that enzyme? And you go through all of those, and that's how you identify these things. You do a series of tests on all of these to determine what they are. And that's what you'll be doing, variations of that in lab, I would anticipate, unless they change the lab. And I did it last year, a year ago this term, though not doing it this term. And I think it's probably the same. And so, and it's based on this, and this is sort of like the Bible, called Berge's Manual of Determinative Bacteriology. I'm not asking you anything about that. It's lab. I don't use those terms, and if they want to use them in lab, it's lab. Okay, so, and this is just definition, species, subspecies, and we'll get into how they, for instance, that serotyping is kind of like basically what I was showing you with E. coli. We're doing tests based on unique surface molecules that we can analyze in the lab to identify them more specifically. So that's what, and we do that all the time, whether we're trying to identify a virus or what's, because it gives us a lot of information. And we're done with that part. Aren't you happy? You don't have to thank me. It's all part of the service. I'll see if there's anything more in the Nestor PowerPoint, just because I had it opened up. Neat stuff. I thought was there. The chromosome, which is called the nucleoid, again, a single circle, really compacted a lot. There's an animation if you're interested, packed tightly by binding proteins and supercoiling. So you have all of this reddish area, and you can see this is one that's dividing normally. Okay, and plasmids differ. They don't encode essential, means required for life, genetic information, similar but smaller, and again, antibiotic resistance, etc., talked about endlessly. When a bacteria dies, this can explode and wrap around that bacteria. It's so supercoiled, it can wrap around 1,000 times. So imagine how tightly compacted, if in a room, if we could encircle this room 1,000 times with some type of coiled rope, string, whatever that was there. Here again, the idea of the 70S, the way they combine, important medically, antibiotics impacting 70S ribosome do not affect the 80S ribosome. That may be true, but again, we have 70S ribosomes in our body, as you will, I'll reinforce when we get to eukaryotic cells, which is next. The cytoskeleton, yeah, probably controlled in cell shape, storage granules, mostly for identification. Because if you do a methylene blue stain, like I said, every so often we'll see these metachromatic granules. When you're looking at Corynebacterium, I'll talk about that again when we get there, etc., etc. Here's the endospores nugget, found everywhere. And sporulation, the process takes about 8 hours. Okay, the endospore layers prevent damage. It doesn't have a lot of internal components. It's got a little bit of cytoplasm, and it's got the genetic material. That's about it. And when it starts to develop, then everything sort of springs from there. It's got some ribosomes in it, so it's got the bare minimum of equipment. That cortex in the dehydrated state protects it. Some more proteins bind and protect it. The dipicolinic acid plays that role, attracting the calcium, extruding the water. And we can make them occur in the lab when in real life, when the conditions are favorable, they grow. That's why when I was telling you about like tetanus, is an anaerobe. So when a wound is a wound, if you see a nasty wound with the skin edges all torn, somebody, really filthy stuff in there, and it starts to turn black around the edges, that's called necrosis. NECROSIS. Necrotic tissue is dead or dying. It doesn't get any oxygen. That's by definition where they can flourish. We look at those and we call them tetanus-prone wounds. I have a really good chapter on wounds that we do toward the end. I have a great outline on wounds. I'm flattering myself, I did it years ago. I know a ton about wounds. For those that don't know, nearly 50 years, I worked in an inner city hospital, and oh my goodness, we saw everything. I don't know if you watch HBO and watch The Pit, or have done so. It's airing now in Pittsburgh. They had a very interesting one in the last episode.
Don't spoil it.
Oh, okay.
I didn't watch it yet.
Okay.
But my mom told me it wasn't even that good, so I guess.
I beg your pardon.
Was it that good?
Which one was that? 3 or 4? 4 was the last one.
4.
Did you see the one with the homeless guy?
Yes.
You see the cast and the homeless guy?
That was 3.
What happened
Yeah.
with the cast?
Oh, there was maggots eating him.
I'm watching that with my wife the other night. Okay, and we buzzed it. Listen, and she says, "Oh, the cast," and they're all looking kind of moving and stuff like that. And I looked at my wife. There's gonna be maggots under there. She goes, "Really?" I said, "I've seen it." They didn't. By the way, they didn't use the cast cutter properly, but I digress. Okay. Oh my gosh, everybody. We had those baths, and they're trying to spray. We had people filled with lice. You had to have delousing inside. Oh, but we were right because we had a tremendously large homeless and indigent population. More on that as it goes on. The experience is agreed, and it's not a means of reproduction. End of sports. Okay, and on to we're going back. I know this is a bit confusing, but that concludes our prokaryote studies on the anatomy, and now we're going to do the eukarya, and some of it will be review. This is Cowan's fourth chapter. That's your textbook chapter, and this is something that's what they call the last common ancestor. We'll get into that. That has to do with something we call endosymbiotic theory. I think that's really significant because it's all about the mitochondria and the 70S. Okay, I'll buy it. I wasn't there despite what you're thinking. I don't really want to talk to anybody. So theoretically, we've all evolved from whoever this ancestor was. Okay, we're likely single-celled and independent. Feels like me. Over time, cells aggregated and formed colonies. I'll buy it. Okay, so I don't even want. I'll never ask you a question about this type of stuff. They became specialized. Multicellular organisms evolved, lost the ability to survive separately from the intact colony. Okay with me. If that's what they're saying, we can buy that. So what happened is, and we'll get to this, the big player here when we look down the road is that it has a circular chromosome like bacteria. There's two big pieces of evidence to support this. That circular chromosome and the fact that they have prokaryotic ribosomes is also part of it. Okay? And it's capable of independent divisions. And this we find is evidence that mitochondria were once prokaryotic cells. They're not all. I mean, I think you probably have to go based on this. You'd have to say all. I don't know if this is as accurate as they would like it to be. So you can read through those. I hate doing that. Voila. That's that. Remember I drew it on the board? That's that. You remember it. 9 plus 2. That is significant. There'll be questions like this. A lot of the questions on the exam will say, in a prokaryotic cell, we see this. In a eukaryotic cell, we see. So it says in eukarya or in a eukaryotic cell, you have to start put your mind. If I'm talking about a flagella or a cilia, that 9 plus 2 is going to come in. If I say in a prokaryotic cell, the flagella, it doesn't have any of that. It's made from flagellin. It's very simple. These are inside the cell. They're intracellular extensions. They're sheathed in membrane. They're just causing the membrane to protrude, and they have a thin layer of membrane, or universally, thin sheath would be the best word, and they protrude. Okay. It's like sticking out. This is an old sweater, who cares? Yeah, here we go. As opposed to in a prokaryotic cell, it's like it's inside, and now it's sticking out like a hair. I did that for you before in A&P. You remember, unfortunately. Same bad stuff. Doing this for a reason to understand the distinctions. They just had on Wednesday the mega pharmacology test, the first one for these students in pharm in the nursing track. I mean, you see boards, all this stuff that I bring to you. So you might think this is not useful. It's going to be useful. So that's the 9 plus 2 arrangement. That is unique to eukaryotic cells. It's one of the things that justifies the concept of evolution, because it has it in these tiny unicellular forms. The flagella of flagellated protozoan, a single-celled organism, It's the same as a flagella and a sperm cell. They look the same. They have the same internal structure. So eukaryotic flagella differ. First of all, they're much larger. They're much thicker, and that has a great deal. That's the complexity. They're covered by the cell membrane, long sheathed cylinder containing regularly spaced microtubules. It's made of microtubules. We don't have those in prokaryotic cells. We don't have intermediate filaments. Okay, the ones that form desmosomes and stuff. The only one they have are microfilaments, those tiny little microactin filaments. Eukaryotic cilia. Bacteria don't even have cilia. If you see the name cilia and bacteria, it's immediately a wrong answer because they don't exist. The term is not part of it. Papillae and fimbriae are smaller proportionately. Eukaryotic cilia, similar to flagella, but smaller and more numerous, found only in single group of protozoan and certain animal cells. They use these to move around. And that is how, in the lab, we classify them. Those things in the pond water, that's their means of motion, the ones that move. So, do they have a glycocalyx? Our cells do. These are like our cells. Direct contact with the environment, polysaccharides, have different kinds of appearances. They can look at fibers. They can make the slime layer. They can have a capsule. The yeast, you're going to be, did you work with yeast in lab this week? Oh, I love yeast. I even bring my own yeast. If you bake or use yeast in baking or cooking, you got to check if it's past that freshness state. That's one of the times when that really matters. It just doesn't give you the what you need. Just saying, I always use more. Why would a little extra yeast wouldn't hurt? Okay, yeast have a capsule. You can do what's called, did you do a negative stain in the lab? Yeast would stain, you see the capsule, you see that blank space around it. Okay, that's one of the things we utilize. It's one of the easy identifiers of yeast. Yeast are, you looked at yeast under high power. You didn't have to use oil. Did you look at them under oil? They're huge. If you look at them under high power, you did oil, yeah. Yeah, fine, does all that. Okay, protozoans, and we're not doing worms, do not have cell walls. Cell walls of fungi, they're really important. They provide a lot of structural support. They're different than those cells' wall. They're made out of chitin, mostly. Sometimes it will be cellulose, but one of the few times, what is chitin? Again, chitin, cellulose, as you might recall, is all glucose. Chitin involves nitrogen-containing chains of carbohydrates. It's the stuff that insect shells are made of. It's the stuff that, like crab leg shells and all that. Those are chitinous exoskeletons. Okay, chitin. So, and that's the part when you're working with mushrooms, cooked culinarily speaking, it's the part you can't eat. Okay, you snap off the stems at the base. Don't throw them away; they're useful for stock. Help me. All right, but chitin is important because the other things don't do that. So, and they're looking at some cross-match. So here you can see them. Mixed glycans, chitin, all of that type of stuff. Just remember that chitin's part of it. But again, these have a cell wall. So if you look back, the cell wall, algae and fungi or yeast have that. What's interesting again about fungi and yeast, they're a hybrid. They do a lot of the activities of our kind of cells, protozoan cells, and even our kinds of cells, in the way that they function, yet they don't have chlorophyll, which algae have. So they're almost like an in-between. Fungi are land-based. Fungi and yeast are land-based. Algae are in water. And protozoans can be either. They can be in the soil or be in the water. So a lot of that. Typical good old phospholipid bilayer, thank goodness. Here's a big player, sterols. The sterol, we have that. Hold on. Message from the boss. When my wife messages me, I have to pay attention. We know that. Okay? So all eukaryotic cells have sterols. We have cholesterol. The only bacteria, that mycoplasma, you remember, they use sterols because they don't have a really well-developed cell wall. Fungi, the only place where we ever see this is ergosterol, called ergosterol. When you start vis-a-vis what they were learning for this first exam, they were learning a lot about antifungals. The target of every antifungal that is used today, other than severe infections, which are having something with a lot of complication, the antifungals target this because it's a unique feature of that. And fungal infections are a big problem, particularly when they get into the respiratory tract. And that's usually in very, very sick people. They target ergosterol synthesis. And you all become very familiar with them, particularly one that affect women very frequently, vaginal yeast infections. They utilize a thing called diflucam, and I can't even tell you how many prescriptions I wrote for that. Diabetic patients on antibiotics frequently get vaginal yeast infections. So moving on. Sterols of various kinds, relative rigidity to the membrane. Important cells do not have a cell wall, but here, fungi that have a cell wall, it's so important. Okay, similar function to those of bacteria, serving as selectively permeable. It's still a selectively permeable. What define why they named eukarya is because they could see the nucleus, and we know it has a nuclear envelope. It has a double layer, a double membrane layer that argues with the specificity and maybe even argues a little bit toward that common ancestor, but I'm not going into it. We talked about the nuclear pores. Remember, nutrients and stuff can come in, RNA can go out, DNA has to stay in, can't get through. Like the dog door and the cat door, right? That. At least the dog is having fun today, if you're not. Dave can't bow out. He's running with the gang. Macromolecules migrate through the pores to cytoplasm and vice versa. Always a test question. Remember I said this before? What happens to the nucleolus? Site for ribosomal RNA synthesis. I have a sneaky suspicion you may see that question again. You're all looking at me. Remember I had that question before? Keep it in mind. It's going to be hard enough, this course. If I'm giving you help, take it. Just like us, they have chromatin, which is DNA that's linear and embedded and basically held in a strand-like position, not supercoiled, in those thin linear ways by those histone proteins, and we call it chromatin. When it gets ready to replicate, we call them chromosomes. The big difference: bacteria replicate by binary fission. They keep just dividing in two, just like you saw in the animation. Here we have mitosis and meiosis. Not all of them undergo meiosis. The simpler they are, the more it's just typically mitosis. Asexual reproduction. Some of them do undergo sexual. Okay. So voila, the nucleus, you saw those, the endomembrane system, you saw that, the nuclear pores. Nothing new on the hit parade for that. Oops, sorry. A series of membrane tunnels used in transport and storage. We knew about that. The rough, because ribosomes bind to it. The smooth, interesting when you, and again, you're going to look at these things and see the importance. They play a role in identification. I do not know if you're doing that in AP2 lab. I'm not. You do a lot of microscopy in AP2 lab? Okay. So we don't have to cover it. And here, we're not going to go into all this and protein synthesis and blah. The Golgi, okay, we went over this. Okay, it basically is a. We won't go into the different ends of it. You don't need to do that. Oh my God. We did all that. Condensing and transitional vesicles. Don't worry about it. Because there's a T side and a C side. And they're all part nature's assembly line. I never heard that term before. Instruction for producing a protein is copied into RNA. And again, you should always remember transcription is DNA becoming RNA, and translation is RNA making proteins. Transcription is always in the nucleus. Translation occurs in the ribosome in bacteria all the time in the cytoplasm. Or in, if they're bound, then it's involved in the endomembrane system. We did all that, don't have to go over that. Lysosomes, you remember? Variety of enzymes involved in the intracellular digestion of food particles and protection against invading microorganisms. So they function in digestion and defense. And vacuoles, particularly, they contain different things. Some they are just storage. Again, mostly for identification. Sometimes there's stuff that's going to be excreted. They're in the process of digestion, etc., etc. They forgot something. I'm not going to go over phagocytosis. We did this before. The one that they should have put in, but they didn't. Now we have to discuss peroxisome. Remember, I commented on that. And the peroxisomes principally contain defensive enzymes to deal with toxic derivatives of oxygen. If it's a species, we'll encounter a number of them. In an oxygen environment, inevitably, oxygen is going to create problems. You get a lot of toxic derivatives of oxygen. Principally, they contain two major enzymes. I'm doing this now because we're going to be doing it again when we start to look at physiology. We categorize them based on their ability to function in the air. Is there too much air? Is there not enough air? Etc. Those two enzymes in the peroxisomes are confusing. SOD, we use the acronym superoxide dismutase. And the other one, you can use this in the lab. You're going to do this test in the lab. It's called the bubble test. The other enzyme is called catalyst. And so, organisms that can survive in an oxygen environment have to either have both of these or at least one of these to be able to function. Effectively, toxic derivatives of oxygen, you might remember, are that O2 with the minus sign, superoxide radical. Free radicals, you might remember, are really the linchpins today of cancer theory, aging theory. And what they are molecules that bump into otherwise stable molecules and destabilize. Part of the expression is biologically equivalent of shit destroyer. It works, doesn't it? It's good. I know. Right away. You have to apologize for bad words if you're like in England on TV on the BBC. They have to apologize if somebody drops a F-bomb. But you have to lose. Nevertheless, superoxide is this free radical. So we have to basically take that O2 with the minus sign, and what we do in the presence, I'll just use the SOD, converts it, okay, I'm not doing the chemistry. It takes me to hydrogen peroxide, okay? Because it adds, we put water in this. So effectively, it converts to hydrogen peroxide as a free radical. It's one of the reasons it works as an anti-infective. There's other ones. The hydrogen peroxide, in the presence of catalase, okay, changes that to water and oxygen gas. That's why when you put hydrogen peroxide on a wound, like us, scratch or scrape to cleanse it, it bubbles. We're going to do that test with bacteria to see if they have it or not. Whenever you get a scrape or a scratch, okay, cells are ruptured or die. They release the catalase in them. If you put the peroxide on them, they bubble and they sting because it's rapid. It's a rapid reaction. If you put it on intact skin, that doesn't have any effect at all. Really important going forward. And you're going to see that plays a pivotal role in understanding it in when we start to look at, there's 5 different categories of oxygen. We are what are called obligate aerobes. We have to have oxygen. Those Clostridium, the C. diff, the tetanus, the botulism, they're obligate anaerobes. Oxygen kills them. It's a good thing. Keep in mind. I'll talk a little bit more about the therapeutics. Perhaps the most important one, and we'll end with this, is the mitochondria. We know it's the energy for the cell. We know that's where all those good things come, and it gobbles up sugars and converts them to scads of ATP, and they have the inner membrane and outer membrane. Okay, the big thing. This is why this thing we call endosymbiotic theory, that common ancestor theory. If I change the name from endosymbiotic to last common ancestor theory, somebody wrote a paper. It means that effectively from this common ancestor, all living cells developed. What's unique about this is mitochondria have their own DNA. It's circular, bacterial DNA. They have their own ribosomes. They're 70S, bacterial ribosomes. So we got, and effectively, when we need more, if you're working out and you haven't done so for a while, all of a sudden your muscles are going to start to make more mitochondria. If you hurt yourself and you have to take some time off from going to the gym or from whatever sport you're doing, the mitochondria will go away. They won't be replicated because they have a lifespan. That's because they have their own DNA. They're like little tiny cells inside. They have an inner membrane and an outer membrane. So they're multiply protected. That's what it is. And so they have circular DNA, bacterial type DNA, and they have bacteria-sized 70S ribosomes. That's why when we target the bacterial ribosome with antibiotics like the 1 zillion of them I saw on the boards down there in Scaife Hall, that's why they cause side effects. Super important. Here you can see it. You can't see it on the electron microscope. You can just see the matrix and the cristae. But here, 70S ribosomes and the DNA molecule, even though they're not drawing it as a circle, it's a circle. That's really important. Chloroplasts, like microplants, algae, photosynthesis, okay, resemble mitochondria but are larger and contain special pigments. If they have chloroplasts, they also have mitochondria. In plants, they have to have both. And the ribosomes, again, are 70S. Nothing special about that. I'm not going to go into anything there, but the typical eukaryotic cell is 80S ribosome, other than the mitochondria, which have their own. And they're effectively the subunits. The largest and most subunits are bigger. And when you add them together, so to speak, functionally, and when they bond together, their density becomes slightly larger at 80S. I will leave it there. And so we don't have a lot more to do with this chapter. We go into the physiology chapter pretty soon, and I'm going to try to get a date set up for next. We come back, what's the 9th? I'm thinking, either the 23rd or the 25th, where this exam is going to be. Fridays are not great days to have it because we have athletes that travel and things like that right now. So start marking that down because someone's going to ask me that question.